This demonstration will show you the proper procedure for preparing and exposing your yeast plate. In this lab activity, 3 groups will prepare 1 plate of yeast each to be exposed to UV light for 30 seconds. Each group will be assigned a different SPF rating to protect their yeast with. First, wash your hands and clean your work area. Plates are easy to contaminate, so we have to be careful. Plug in and turn on your UV source in order to allow the bulb to start warming up. Do not look or put any body parts under the panels covering the UV box while the light is turned on. UV radiation can cause serious eye damage and skin cancer. Obtain a Petri plate of sterilized media. Make sure to keep the plate closed and upside down as often as possible to prevent microbes in the air from falling into your plate and condensation from dripping from the lid onto your agar. Use a permanent marker to label the bottom of the Petri plate with your class time and day and the SPF your group was assigned. You will use a sterile swab to spread some yeast on your plate. Make sure to open the correct end of the package containing the swab so that you don't contaminate the end that you are going to work with. Dip your swab into the yeast suspension and then open up the Petri plate and gently rub the swab back and forth along the agar, rotating the plate as necessary to make sure that you have covered the whole area with yeast. Close your plate and set it upside down. Be sure to dispose of your swab in the appropriate waste container. Cut a piece of plastic large enough to cover the top of the Petri plate. Turn your plate right side up and drape the plastic wrap over the lid. Your plate should still be closed at this time. Be sure that your plastic wrap is as flat and as spread out as possible so that you do not have creases that the sunscreen can't get into. Trace the edges of the plate onto the plastic wrap using a permanent marker. Spread a very small amount of sunscreen with your assigned SPF evenly over the plastic wrap using a sterile swab or with your finger. Be sure to spread the sunscreen well. You should not be able to see any sunscreen on the plastic wrap when you are finished. Remove the plastic wrap from the Petri plate lid. Open the lid and drape the saran over the open plate. Pull it tight around the sides to keep it from hanging down and touching the surface of the agar, or you will contaminate the plate. Turn off the UV source and place the Petri plate inside. Remember, do not look or put any body parts under the panels covering the UV box while the light is turned on. Now, using a watch or clock in the lab to time yourself, turn on the UV source and expose the plate for 30 seconds. When 30 seconds has elapsed, turn off the light source and remove your plate. Now you can carefully remove the plastic wrap, making sure that it does not touch the agar. Replace the lid and turn the plate upside down. You are ready to turn this in to your TA. The plates will be incubated upside down at 30 degrees Celsius for 2 to 3 days to allow the yeast to grow, and you will view your plates during the following class meeting. But for now, we can look at some already prepared plates to see what result you should expect to see in class. We would expect that the more UV the yeast are exposed to, the fewer of them will survive because UV damages DNA. We would also expect that a higher SPF rating would provide a stronger shield against the UV and therefore would protect the yeast better. So we should see less cell death as the SPF rating goes up. If we prepare a yeast plate and don't expose it to any UV, then we should get a full lawn of bacteria, which means that the plate should be completely covered with growth and will look like this. If we prepare a plate and expose it to UV for 30 seconds without any sunscreen, we can see that the radiation kills off a large amount of yeast by creating mutations in the cells that cause them to die. So we can see that there is a big difference in how healthy the yeast are when they are exposed to UV and when they are not exposed to UV. The non-exposed have survived and grown much better. Remember, the sunscreen is going to act as a shield to block the UV rays from hitting the yeast. So we should see them survive better and better as we apply stronger shields. If we expose 3 more plates, for 30 seconds each, protected with sunscreens with increasing SPF ratings, we can compare how well these shields protect against UV based on how much growth they allow. This plate was protected with SPF 5, and we can see that SPF 5 protects better than using no sunscreen because there is more growth on the SPF 5 plate than on the plate that was exposed but was not protected. This plate was protected with SPF 15, and we can see that SPF 15 allows significantly more growth than SPF 5 when we compare the plates, and this makes sense because it is a stronger shield. This plate was protected with SPF 45, and we can see that SPF 45 protects significantly better than SPF 15. These results are what should be expected based on what we know about the effects of UV radiation on DNA, and what we know about how different SPF sunscreens should protect us against UV. It has been said that sunscreens with SPF ratings greater than 15 do not give you any additional protection. Based on the results of this experiment, do you think that this is true? Which SPF would you choose?